Great pleasure to welcome you all to this first seminar of the QBI SBI Seminar Series. This is actually a result of the COVID pandemic. We have trying to organize some meetings for about 2 years now. We always our best efforts were also coupled by COVID, and we managed to have a virtual meeting last year. But in order to continue the interactions, we thought we'll start the seminar series, and it's a really great pleasure to have Danielle Swaney as the first speaker of the seminar series. Danielle did her PhD training at the University of Wisconsin and focusing on proteomics, and this is still what she has been pursuing, and is now working at QBI, also doing a lot of proteomics. And I was actually just talking about this. There's 3 back-to-back papers from QBI in the latest issue of Science, all about protein-protein interaction networks. And Danielle said she will be talking about some of these results. So we are looking very much forward to that. And without further ado, I'll hand over to Danielle.
Great. Thank you very much for the invitation to talk today. Yeah, we're really excited. We had these 3 papers published in Science 2 weeks ago. And so I'm going to tell you about one of the common themes throughout these 3 papers, which are new functions of PIK3CA signaling in cancer. And this work was all part of this cancer cell map initiative that started at UCSF and UC San Diego about 7 years ago now. And the Cancer Cell Map Initiative is a collaborative network of approximately 10 different labs. It was started by Trey Eidiger at UC San Diego and Nevin Krogan here at UCSF. And they brought together these 10 different scientists to get NIH funding to study network biology of cancer. And along the way, we also kind of inherited Natalia Jura, who has great expertise in receptor tyrosine kinases and PIK3CA, which is really, really critical to understanding some of the biology we're discovering here. And so, you know, one of the motivating factors behind the Cancer Cell Map Initiative was to really try to view cancer through a network biology lens, because we think that's a really powerful way of looking at it. And so with that in mind, a primary goal was to build cancer-specific protein networks, so physical and functional interaction networks that we could then take this vast amount of patient DNA sequencing that we don't always know what it means and how these different altered proteins and genes fit together and work together, and try to view them through the lens of these networks to then translate this, you know, vast amount of patient sequencing DNA into predictions or understandings of patient survival, or what sort of drugs might be effective for different patients based on their collection of alterations. And so that's really the motivation behind what we're doing. And, you know, one might say, well, don't we have like a lot of information already about what networks exist in the human cell? And it's true, we have a lot of information. You know, there have been tremendous efforts, you know, tens of millions of dollars invested in discovering protein-protein interactions in human cell lines. But a lot of these interactions are derived from workhorse cell lines. So for example, you know, the heroic efforts of the Bioplex at Harvard has cataloged almost 120,000 interactions, but these have been done in HEC293 T cells just because it's a very tractable cell line. Other efforts by Matthias Mann's lab has used APMS in HeLa cells and derived thousands of interactions there as well. But one thing I think that was really brought to light as the Bioplex expanded upon beyond HEC293 T cells is that when they moved into this colon cancer cell line HTC116, now you're starting to see that there's only a moderate degree of overlap in the protein interactions that are identified in these 2 cell lines. And there's a lot of space here, which are interactions that are unique to this colon cancer cell line. And so I think this really brings to light or emphasizes that there is a vast amount of information about protein-protein interactions that is left to be discovered, because we simply haven't looked in the cellular context in which that interaction exists. And many of these may be relevant to understanding cancer biology. And I think a really nice example of the power of this is actually from your lab, Walter. Last year, you had this great paper out where they did a protein interaction analysis of the entire EGFR network, and looking at this in the context of different KRAS mutations. And I think a really interesting finding from that is, from these dozens of baits that they profiled, when they have different KRAS mutations, you see different levels of interaction rewiring. So for some proteins here, they have many mutations, more than half of them are rewired, which are the red and blue here, when you're in different cellular contexts, whereas other proteins are identical no matter what the cellular context of KRAS is. And so this really suggests that, the cellular context is important. And what's more important actually is that when you know this rewiring and you understand the mutations that underlie these cellular contexts, then they show that you can actually start to stratify patients into those with higher or lower survival. And this is really a great illustration of what the motivation of the Cancer Cell Map Initiative is, to be able to use these networks, how they're rewired, to understand patient data. And so that's what we're trying to do here. And so what we really want to do is build more extensive physical and functional interaction networks. And so the high-level approach that we've been using to do this is affinity purification mass spectrometry. And for the work I'm going to be talking about today, this really focused on either proteins that are altered in head and neck cancer or breast cancer. And we select many of the top altered genes from these cancers and performed protein interaction analysis using APMS in both normal cell lines that are just immortalized cell lines from these anatomical regions, as well as in tumor lines that represent these types of cancers. And then we can identify protein-protein interaction and build networks. And to complement this work, what we also did, and so I led the work head and neck cancer, and my colleague Min-Kyu Kim led the work on breast cancer. And then to complement this, our colleague at UCSD, Fan Zhang, also brought in a huge amount of public database information that can inform PPIs. This included physical interactions like the BioGrid, BioPlex, other things like protein co-expression, which has been shown to be useful in understanding physical interactions, and brought all of this together into a network of genes relevant to cancer. And so what I'm going to be talking about in this talk today is 3 little stories that kind of came out of this work, all focused on PIK3CA signaling. So identifying novel PIK3CA interactions, new negative regulators of PIK3CA, and then some therapeutic targets for PIK3CA that came out of this work. But to start, I want to just go talk about some of the things that came out of this work that Fan led, utilizing public databases in addition to what we were identifying with our new interactions from head and neck cancer and breast cancer. And one of the PIK3CA related stories that came out of that related to the actomyosin complex. So what Fan was really doing was he was asking the question, what is the right network scale for interpreting cancer data? You could be looking at just binary protein-protein interactions, small complexes, large complexes, whole organelles. What is that? So if you take a look at just what the alteration frequency is for a given cancer type, here's colorectal cancer, you see commonly altered proteins such as APC, KRAS, for example, have high levels of mutation, and then we have this long tail of very low levels of interactions. And so what Fan did was to using public information about how these genes are connected at a physical scale, organize them into protein complexes, supercomplexes, organelles, into this kind of hierarchical structure of different protein systems, as he's termed it. And then he asked the questions, what is the rate of mutation within these different systems, whether it's at a small level of, binary protein complexes or larger super complexes. And then, look at these systems at all these different levels. What is the frequency of alteration at these systems? And here you can see that, there are a lot more things with high levels of alterations as we organize them into pathways and higher order complexes. And so, one of the questions that we then asked is, where do we see convergence of mutations? Where is there a higher level of mutations than one would expect in these systems? And one of the examples that came out of this analysis was the PIK3CA actomyosin complex. So here's an image of what this complex looks like. And you can see the lines in between different components of the complex illustrate what type of data it was coming from. So there were, the red lines are physical interaction data, what that was present in public databases. We have some instances of sequence similarity, for example, in this teal color. And then what you'll notice here is, this is a highly interconnected actomyosin complex. But then our new information from head and neck cancer in particular implicated PIK3CA as interacting with multiple known components of this actomyosin complex. And so this was potentially a new regulator of actomyosin. So to try to see if there was, so just a little bit about PIK3CA signaling. So PIK3CA is a lipid kinase. It takes signals from the from outside the cell, and then its kinase activity converts PIP2 to PIP3, which can activate downstream signaling such as AKT, which promotes cell proliferation. And this process that PI3 kinase mediates from PIP2 to PIP3 is then the reverse process is mediated by PTEN. So PTEN is a negative regulator of PIK3CA. And what we observe when we look at the alteration levels of this new actomyosin PIK3CA complex is that, as expected, PIK3CA is a very well-known oncogene in cancer in general, and depending on different cancer types you look at, breast cancer, head and neck cancer, bladder cancer, PIK3CA is altered at high levels in all of these cancers. But then popping up at the second most commonly altered proteins in this complex here are these new components that PIK3CA is connecting to, myosin 9 and myosin 10. So we wanted to see, is there a functional connection between PIK3CA and myosin 9 and 10? And so we looked to the TCGA evidence for data. And here we performed a mutual exclusivity analysis. And because oftentimes if you have genes that are rarely mutated at the same time, this indicates that they have a functional redundancy. And so what we observed here is that there is indeed mutual exclusivity between PIK3CA and myosin 9. And then if we look at the RPPA data from the TCGA, which is a protein-based array, two of the components of that array measure downstream PIK3CA signaling via AKT phosphorylation at threonine 308 and serine 473. And what you can see here is that if we look at myosin 9, for example, when you have mutations in myosin 9, we see higher levels of activation of downstream PIK3CA signaling as compared to wild type myosin 9. And the same thing is true for myosin 10, where we see increased levels when we have a mutated version of this gene. So this really got us thinking about a model such as this, where normally PIK3CA is promoting downstream activation of AKT, but when we have actomyosin, this can potentially sequester PIK3CA and then inhibit downstream signaling. So a way of inhibiting PIK3CA signaling. And to try to test this more systematically, what we did is we performed an experiment in a cell line where we observed this interaction between PIK3CA and actomyosin, which is Cal33, a head and neck cancer cell line. And what we did is we treated it either with blebbistatin, which can disrupt this actomyosin, or alpelisib, which is a PIK3CA inhibitor. And when we treat it with blebostatin, we see an increased level of phosphorylation of two downstream components of PIK3CA signaling, both phospho-S6 and phospho-AKT. And meanwhile, if we do this blebostatin and the PIK3CA inhibition, we see no such increase in phosphorylation status. So this really suggests that myosin, the actomyosin complex, can sequester PIK3CA, and when we disrupt actomyosin, that releases PIK3CA and enables it to then promote downstream signaling. And in further support of this model, we did this exact same experiment in a different cell line where we don't observe any interaction between actomyosin and PIK3CA, and we see no increase in phosphorylation signaling if we disrupt actomyosin. So this really suggests that the potential model for what's going on is that when PIK3CA can interact with actomyosin complexes, which are comprised of myosin 9 and myosin 10. However, when you get mutation to these two genes, then this can disrupt that interaction, allowing PIK3CA to then promote downstream signaling. So mutations to myosin 9 and myosin 10 are another way for the cell to activate PIK3CA signaling, which is something it desperately wants to do. So that's one little story. Another finding about PIK3CA that we observed was a novel PIK3CA interaction. And this is coming out of the new data that we collected for this paper. So I just want to take you through what we did to collect this new data. So here we had two different cancer cell types. We had head and neck cancer, and we used two different head and neck cancer cell lines, or a non-tumorigenic esophageal cell line Het1A, and then the same sort of experimental design for breast cancer. We had two different breast cancer cell lines, and then a non-tumorigenic breast epithelial cell line, MCF10A. And so in each one of these cell lines, we expressed dozens of genes in biological triplicate, and all of these genes were fusion constructs. So they had a 3x flag tag attached to them. And then we use affinity purification mass spectrometry to purify these protein complexes, identify what are our high confidence interactions using COMPASS and SAINT. And then also in many cases, we included both wild type as well as mutant versions of some of these genes. And so then we perform a label-free quantitation to look at how interactions are precisely regulated by their mutations. And so from that, we identified these networks of protein interactions in head and neck cancer and breast cancer, respectively. And one thing that we observed immediately from this data is that the vast majority of these interactions, 81% of them, are novel. They're not found when you look at, many different public databases. And this, I think, is not necessarily surprising based off of what has been observed with Bioplex endeavors, where they also see a high degree of novel interactions. Additionally, when we look at the overlap between baits that were analyzed in the same cell line, so for example, if we just look at this in head and neck cancer cell lines, we see there's a low degree of overlap between these new interactions. And this is really supported by this novel interaction percentage, where different cellular contexts allow for different interactions to occur. And so one thing we're really interested in doing is comparing interactions between one cell line to another cell line and looking for ones that may be, found in both of our cancer cell lines, but not in our non-tumorigenic cell lines, because these may be interactions that are more conserved in cancer and would be good therapeutic targets. But one thing we realized when we tried to do this is we didn't have such a nice elegant design as Walter had done for his EGFR signaling, and it was difficult to compare protein interactions across cell lines, because normally when you do these types of experiments, you have a protein interaction cell line list in one cell line and you order it in decreasing confidence, and then you apply a cutoff, maybe a 1%, a 5% false discovery rate of interaction detection. And then if you want to go do that in the second cell line, basically you'll end up with two different lists of high confidence interactions. And if you find an interesting interaction in one cell line, then you want to look, is that in the other cell line? And if it's just below the threshold, you may make the false assumption that this interaction doesn't exist in cell line 2, but in fact it exists. We just falls right below the cutoff, or it may be that it's way down the list or not even present at all. And those are very different scenarios. So we wanted to have a more quantitative and statistical way of comparing interaction between cell lines. So we developed a differential interaction scoring to do this type of comparison. And you can use this differential interaction scoring in a lot of different ways. But the way that we prioritized using it was looking for interactions that were conserved across our cancer cell lines and absent from our non-tumorigenic cell lines. So here, basically what we do is we take the confidence score of that interaction in our 2 cancer cell lines and multiply those together, and then multiply that by 1 minus the confidence score of our non-tumorigenic cell line. And this will give us basically the confidence that this is a cancer-enriched interaction or a cancer-depleted interaction. And we've actually used this previously in a paper last year studying coronavirus, where instead of comparing across different cell lines, here we compared across different pathogens, SARS-CoV-2, SARS-CoV-1, and MERS, which all have baits that are highly similar between those different pathogens. So what does this look like in practice? So basically, for an example like CDK5, let's take CDK2 here. In the pie chart here, we have what the interaction confidence was in all 3 cell lines. So the 2 cancer cell lines are in blue and purple, and in green is the non-tumorigenic cell line. You can see it's a dark color in all of these, meaning CDK2 was recovered as an interaction partner in all 3 cell lines, and it gets a gray line here. Other interactions like CDK4 seem to be exclusive or enriched in the cancer cell lines, and so it gets a pink line for a head and neck cancer enriched. And then those interactions that seem to be depleted from head and neck cancer and observed primarily in HET1A non-tumorigenic cell lines are shown in teal here. And one interesting interaction that popped out of this was an interaction between 2 of the baits in our study, PIK3CA, which is highly altered in head and neck cancer, and CCND1, which is also activated extensively in head and neck cancer. And actually, we're really interested in this interaction because if you just look at the combined alteration frequency between PIK3CA and CCND1, almost half of head and neck cancer patients have an alteration in either of these genes. So it's very prevalent. And so we wondered what was going on here. And one of the reasons this is kind of a peculiar interaction is normally CCND1, this is a cyclin, is present in the nucleus to help regulate cell cycle. And PIK3CA is normally present, at the plasma membrane and the cytoplasm. And so one of these proteins is obviously not in its normal localization. And there's evidence in literature for either of them being in the opposite location, but we want to determine, is this a real interaction and where is it? So we performed a proximity ligation assay to try to validate this interaction, identify where it was from. So we use the same cell lines that we use for protein interaction analysis. And then we use anti-flag antibodies to look for CCND1 and anti-PIK3CA antibodies to look for PIK3CA. And so what's shown here in this microscopy figure is in green, we just have a cell mask to kind of show where the cells are. In blue is a DAPI stain of the nucleus. And all of these white dots here are where the interaction is happening between CCND1 and PIK3CA. And when we look at what the localization of those interactions are, we can find that they're predominantly 85% or so found in the cytoplasm. So this suggests a model where CCND1 is leaving the nucleus and interacting with the PI3 kinase complex out in the plasma membrane. And one might hypothesize that this could then impact cell proliferation. And so then we just compared what is the proliferation rates between the cancer cell lines we looked at, between the 3 cell lines we evaluated in this experiment. And this is just correlation, but we find that the HET1A cells where we observe CCND1 interaction to be in the cytoplasm here has the lowest proliferation rate as compared to our 2 cancer cell lines. So I think this is a really interesting complex that really warrants further investigation to understand what CCND1 is doing out of the nucleus. So next, I want to move on to one of the interactions that we detected in breast cancer, looking at a negative regulator of PIK3CA. So as I've discussed before, PIK3CA has downstream signaling to activate AKT phosphorylation. And in breast cancer, here is the map of mutations present in PIK3CA in breast cancer. The predominant mutations are in the kinase domain region, H1047RLY mutation, but there's also a prevalent number of helical domain mutations in the E545 position. So we performed APMS experiments to look at how protein interactions are regulated between wild type and these 2 prevalent mutants in PIK3CA. And from this analysis, we identified 4 known interactions of PIK3CA, its regulatory components PIK3R1, 2, and 3, as well as IRS1. And we find that IRS1 is that interaction is dramatically increased when you have different mutations of PIK3CA. And then we also identified the majority of the interactions identified were actually novel PIK3CA interacting partners, which hadn't been described in public databases before. And we find that in many instances here, these interactions are lost. These interactions are present with wild type PIK3CA, but lost in the mutant. And so we thought this was really interesting. And so we wanted to see, can these regulate PIK3CA function? So we performed an experiment where we evaluated phospho-AKT signaling when we knocked down different interaction partners that we observed in our experiments. And as control, we have some non-targeting controls. We also have P10, which is the negative regulator of PI3K signaling. So this is, a positive control for activity. And we find that in several instances, we see a statistically significant increase in AKT activity when we knock down some of these genes. And this increase is equivalent to knocking down the canonical negative regulator of PIK3CA signaling. So then we wanted to understand some of these a little bit better. So we took the top 2 proteins that seem to be regulating a downstream AKT signaling and performed in vitro kinase activities assays. So here we basically have PIK3CA and in a test tube, either wild type or 2 different mutants of PIK3CA, as well as these 2 different proteins, either recombinant SCGB2A1 or recombinant BPIFA1. And what we observe is that when you have these proteins in an in vitro kinase assay, the kinase activity of PI3 kinase really depends on what the mutational status of this kinase is. So with wild type PIK3CA being dramatically inhibited by the presence of these proteins, while the mutants are less inhibited or not inhibited at all by the presence of these 2 proteins. So this really suggests a model that when you have wild type PIK3CA and these 2 negative regulators present, this inhibits downstream AKT signaling. But when you have mutations to PIK3CA, this allows it to escape this inhibition and allowing PIK3CA to then not be sequestered by these proteins anymore and promote downstream AKT signaling. So this is a way that mutation of PIK3CA can release it from this negative regulation. Okay, so the last story that I just wanted to tell you about is one of the interactions that we observed that we think is a useful therapeutic target. And this was coming out of the head and neck cancer work. So here is a chart looking at what are the protein interactions that, or excuse me, what are the mutations that are present in PIK3CA in the head and neck cancer TCGA data. And here you can see it's a little bit different than breast cancer. In head and neck cancer, the predominant interactions are in this helical domain at positions E542 and E545, and they're present at about 2.5 to 3-fold the frequency as the kinase domain mutations present at a 1047R. And there are several other different mutations present along the PIK3CA gene. And what's interesting about these mutations is they don't all localize to one part of the protein. They're found all over the 3-dimensional shape of PIK3CA. So they have a lot of potential to regulate this kinase activity in many different ways. And so we performed an analysis of all 15 of these different mutants that are present in head and neck cancer patients and looked at how they regulate their interaction partners in one of our head and neck cancer cell lines, SCC25. And what's shown here is all of these different mutants and their interaction partners on the top. And one thing that really is striking here is this box over here, which in red are interactions that are gained by different mutants. And what we find is that these helical domain mutations, which are the most prevalent mutations found in head and neck cancer patients, have a dramatic increase in several proteins over here. And so a little bit about, how PIK3CA is activated. So normally PIK3CA is in an inactive form due to autoinhibition by the regulatory subunit P85. So PIK3CA encodes for P110 alpha, the protein P110 alpha, and then it functions with P85, its regulatory subunit, which is an autoinhibition. And to activate PIK3CA, you really need two things. It needs to go to the membrane, and then you need to remove this autoinhibition of the P85 by having these SH2 domains of P85 interact with phosphorylated tyrosine residues. So when you have some mutations of PIK3CA, such as the kinase domain mutation H1074R, this provides an inherent ability now of PIK3CA to localize to the membrane without the assistance of other proteins. And then it has, the first box here checked, and then all it has to do is interact with some phosphorylated tyrosines, preferentially this YXXM motif present on many proteins, and that can activate PIK3CA. But other mutations of PIK3CA, they can release that autoinhibition of the SH2, the P85 regulatory subunit, but they don't have that intrinsic power to get to the membrane and stay there. So they have to rely on other proteins like HER3 to localize to the membrane and then utilize the density of these phosphorylation events on the tail of HER3 to activate PI3 kinase. And so we hypothesize that this, membrane localization and the density of these activating YXXM motifs on HER3 may be why we observe this dramatic increase in interaction between helical domain mutations on PIK3CA and the HER3 receptor tyrosine kinase. And so this also doesn't seem to be a coincidence. If you look at the TCGA data and we look at HER3 and the activator of HER3, which is neuregulin, so shown here is in green is the levels of neuregulin mRNA. And you can see that across the TCGA pan-cancer atlas studies, neuregulin levels are much higher in head and neck cancer than in any other cancer type profile. So this really means that there is, the gas pedal is on for HER3. Neuregulin is turning on that protein and inducing a lot of phosphorylation in head and neck cancers. And so this really primes HER3. It probably has a highly phosphorylated state, and it's really basically asking for PIK3CA to come interact with it and promote downstream kinase signaling. And additionally, like I mentioned before, head and neck cancers have a preference for this helical domain mutation. And so we hypothesized that there's this synergy between HER3 and PIK3CA helical domain mutations. And if we can just inhibit HER3, we can reduce the phosphorylation status of its internal tail. This would then inhibit the interaction of PIK3CA with HER3 and hopefully stop or inhibit tumor progression. So to test this in vivo, we used some xenograft models, isogenic models, where we had either a mutant 1047R PIK3CA, which we observed to have a low level of interaction with HER3, and then 2 of the helical domain mutations, which have a high level of HER3 interactions. And what we find is that for H1047R, which can localize to the membrane by itself and has low levels of HER3 interactions, it doesn't care if you inhibit HER3 or not, it just keeps growing, completely resistant to HER3 inhibition, which is in red as compared to just normal saline control. However, if you have mutations to PIK3CA that are have high levels of HER3 signaling, they're very dependent on HER3 interactions, such that if you inhibit HER3, now you completely inhibit tumor growth over the course of 2 weeks of treatment with this HER3 inhibitor CDX3379 for either of the helical domain mutations. And so this really suggests that potentially in clinical trials of this inhibitor, patient selection based off of PIK3CA mutational status could be a really important factor to consider when we're deciding whether a patient should get this treatment or whether we would expect them to have a positive outcome from such treatment. So I'm going to stop there and just summarize. So from these 3 different papers, we've identified several new functions of PIK3CA where the actomyosin complex can sequester PIK3CA to inhibit its signaling, and mutations in the actomyosin complex then release that sequestration of PIK3CA, enable it to promote AKT signaling. Another negative regulator of PIK3CA, BPIFA1 and SGB2A1, which seem to have specificity for regulating wild type PIK3CA, but mutant PIK3CA can escape this this negative regulator. We identified novel interactions of PIK3CA with CCND1, placing CCND1 in a non-canonical localization of the cytoplasm, and then looked at how different PIK3CA mutations and their interaction partners can then be deterministic in the response to therapeutic treatment. So with that, I'd just like to acknowledge everyone who worked on this project, the CCMI team leads, our funding from UCSF and the NIH. So I'm affiliated with the Krogan Lab, so all of the members of the Krogan Lab have listened to this work for a long time, and I really appreciate all their feedback. And then the stories that I told today were focused on were 3 different were out of these 3 different papers that came out in Science 2 weeks ago. There's much more beyond the PIK3CA stories that I chose to focus on today. Also finding new interactions, for example, for BRCA1 in breast cancer and how it can be regulated by these new interactions. So I'd be happy to take any questions if there are any.
Yeah, thanks a lot, Danielle, for a really fantastic talk. I really enjoyed it, and I think everybody else did as well. It's just enormously fascinating data about the organization of a cell and what can go wrong. So please put your questions into the chat or into the Q&A function. I'm going to be looking at both. But maybe can I ask the first question? There is quite a tissue specificity in many mutations. So for instance, a mutation in BRCA1 will be specifically causing cancer in breast and in ovary, but not in other tissues. Is this reflected in this protein interaction changes? Can this explain in part tissue specificity of mutations?
I think Potentially, yes. I don't think we have enough data to, really look at that across different tissue types yet. I mean, we only, between like, for example, the head and neck cancer and breast cancer data sets that we collected, there were only a handful of proteins, including PIK3CA, TP53, that were characterized in both. And, for example, HER3 wasn't found to be an interaction partner in breast cancer at all, even though, it has been targeted therapeutically in that cancer type.
Yeah, this actually because the HER2, HER3 dimer seems to be really sort of the worst of the HER family genes in breast cancer. And I was actually wondering whether the therapeutic antibodies like Herceptin would influence that interaction, but since you're saying it's not there, it's but these different mechanisms which you showed for sequestration or regulation of PI3 kinase, do they coexist in cells, or do separate cell types use separate mechanisms?
Yeah, most
the sequestration versus the association with inhibitors were.
Yeah, so for example, the breast cancer negative regulators that I described, those ones we did test in other cell lines, I believe, and found that to exist in other cell lines. So we don't know the degree of how applicable these are across different cellular contexts, but I think some of them definitely will be.
Okay. There is a question from Callum Ryan who says, amazing talk. Have you any ideas how we can scale this to all of the other cancer types? Do we actually need experimentally derived maps for each, and maybe for each subtype?
I mean, I would hope we don't need to get experimentally because that's a lot of work. I think one thing that we definitely need are more high throughput approaches to monitor protein interactions. So I think that's where, from a proteomics standpoint, cross-linking approaches where you can look at interactions at a whole cell in a single experiment, or some of these nice biochemical fractionation experiments to look at protein interactions are useful, but also other technologies beyond proteomics that might be applicable here to help look at these. And also there's been great work, looking at co-expression of proteins to help also elucidate or infer protein complexes. And this can be really useful and also high throughput. I think people like Willie Haas at UMass in Massachusetts has done a really great job of showing how this can be useful as well.
Okay. George Oliviero has a question: how do protein isoforms affect the protein-protein interaction landscape, in particular localization in different cellular compartments?
Yeah, that's a great question, and I don't know the answer to that. I mean, I've mostly focused on just canonical isoforms here, but certainly there's a whole new potential mode of regulation there.
Yeah, I mean, there's particular in cancers, there's a lot of alternative splicing going on,
Exactly.
Which would produce probably quite a different landscape in isoforms than you have in normal cells.
Yeah.
I think this was all the questions. If there is any more, please put it in the chat, but be quick. I would like to thank Danielle for a really fantastic talk. It was great to have you speaking at this opening seminar. And before you all leave, please just allow me to show one quick slide. And this is about, can you see that?
Yeah, you can see it.
It's about a postdoctoral program which is funded by the European Union. It's called DevelopMed, and this is particular for postdocs who are coming from abroad into Ireland and want to do a postdoc in the broad area of precision oncology. The application is currently open, and Callum Ryan, Jonathan Bond, and myself from SBI are potential supervisors. So if you feel like Ireland would be a good place to do a postdoc, then please apply. The application information is here, development@ucd.ie. And the application deadline is end of November. So thank you very much, everybody, for listening and dialing in. And again, thanks, Danielle, for delivering such a great talk. It's a good way to begin the weekend, for us, at least in Europe.
Thank you. My pleasure.
Thank you. Bye-bye.
Bye.